Alright, we're here today to talk about protein synthesis. We had it done for today, but the problem is somehow it corrupted when I was uploading it to YouTube. Don't really know what happened. Not really going to question it right now. So Sam and I are actually going to finish this tonight and get it hopefully a decent grade on our 10 points. Um, but I'm going to quick talk about protein synthesis as like a basic summary. It's basically one of the most fundamental biologic processes um, by which like individual cells perform. Um, it involves DNA, RNA, and basically a, almost every part of the cell. Maybe not the cell, but like the ways of creating new things like RNA, DNA,
Ribosomes.
ribosomes, and enzymes. Um, and we're specifically going to talk about transcription and translation. And I'll actually pass that over to Sam.
All right, so transcription falls into three stages. The first one being initiation, the second being chain elongation, and the third being termination. The first stage occurs when the promoter sequence, which is the first row of nucleotides, and this is where it's just starting it all off, and that's just basically what initiation is. Next, in chain elongation, that's where the RNA polymerase actually reads the template DNA. So this is when the like only one strand of the DNA is read for the base sequence, and the RNA is completely synthesized with the complementary strand of DNA. After that, we have termination, which this is actually where the whole process is done. It's also after, right at the end of transcription, is where they add a poly A tail to prevent the digestive enzymes in the cell to break down the RNA recipe. And that pretty much sums up transcription.
Alrighty. And then I'll talk about translation. Um, this is when mRNA like starts contains the information needed to like synthesize protein. Starts like sending messages to like the cytoplasm and the ribosomes, and trying to get them to gather up and start the process of actually creating a protein. Um, the mRNA can be like thousands of nucleotides long. It's not just like one or two or like 100. It can literally be thousands. Um, in like eukaryotes, there are only like single coding regions on each RNA strand. But in like another example as prokaryotes, um, they're more like coding one region that presents like the same RNA. Then the main objective of translation is really to convert the genetic material in the mRNA to like a oh my god, functional protein rather than just like it being information that needs to um, pass on to a different part of the cell and making it actually do something more functional for the cell as a whole. Um, like proteins, peptides, and polypeptides are used like interchangeably to describe um, like proteins, I guess, and like how it's made throughout the process. Um, there's another kind of RNA, the tRNA, which finds the amino acids and carries them to the ribosome to start creating the whole like the protein, so it can actually create what it needs to. Um, the codons that are created, it really depends on what kind you want, but the cell sends the message to the mRNA, and then the mRNA passes on to the tRNA. And when it's finding the amino acids, it can find the specific anticodons that it needs for the sequence. There's a possibility for like a mutation where It actually deletes or replaces and possibly even adds another nucleotide when it is not needed. This can cause major problems, like causing like cancer suppressors to not do their job correctly and actually let your body start to mutate and cause more like cells to actually like hurt your body rather than help it advance so you can live more. A little bit more on mRNA is it's the source of the coding information for protein synthesis. It helps basically do everything for the whole process. It is the initiation. It tells like the tRNA about the elongation it needs to do, and it ends up terminating the whole thing in the end. And
Right.
that's about
Oh,
it.
well, here I got something else to add.
All right.
So that's generally the way that protein synthesis goes about, but there are things called protein precursors or proteins or propeptides, which are actually inactive protein that can be turned on like at any moment. And these proteins are used to help with like when the body needs something right away. It's short notice. And an example of these are the enzymes of digestive tracts in humans because they're needed to react faster than the whole translation transcription process can make it. And yeah,
Um, yeah. And like this whole process, it seems like it might take like in like it seems like it might take like a day to make all of this, but in truth, it takes actually maybe a couple minutes to make a protein. Maybe even shorter. It depends on how big the protein is and how fast the body needs it. As Sam was saying about the like digestive proteins and stuff, things like that. I think that might be it.
yeah, that pretty much sums up protein synthesis.
All right. Well, thank you, Mr. Boland, and other people if they listen to this for actually listening to this, and thank you.